I thought I'd just give you a brief overview of cholangiocarcinoma from a kind of Edinburgh and Glasgow perspective. We sit up here, far above the boundary with the Picts, and we've been kind of working on cholangiocarcinoma for about, kind of research active in cholangiocarcinoma for about kind of 7 years or so. And this isn't data from us, but this was published last year. Scotland actually does pretty badly for cholangiocarcinoma, even compared to the rest of the UK. So in men and in women, the rates of cholangiocarcinoma are higher typically than in England, certainly, and roughly equivalent to what you see in Wales. And the net survival is actually worse, and it seems to be getting worse. And there's a kind of question mark, I think, about why that is in Scotland. There are obviously socioeconomic factors, there are potentially genetic factors in Scotland that kind of contribute to these trends. So our approach to tackling cholangiocarcinoma really has been this. So we are a collection of people who enjoy basic research and utilize basic research to understand fundamental mechanisms. We then try to move that through both trials and also kind of industry collaborations into a kind of translational setting with the ultimate aim that this will kind of go into clinical practice. And we're a reasonably jolly team of people. So at this end, you've got kind of scientists, so Ben, who is now in Australia, and myself, Stuart, who sits somewhere at this kind of interface, as does Ian Tomlinson, and then our kind of surgical colleagues, Steve Wigmore and Rachel Guest. But what we should also note is that Rachel feeds very much into the science, as does Stuart, and Steve doesn't do so much anymore, but is very important. Not to miss, obviously, Tim Kendall, who's in the audience, and as we discussed yesterday, is our reason for having some high-impact papers, because he's a pathologist. And over the last few years, so really since 2014, I think we've been making some pretty good headway in identifying new, potentially therapeutic mechanisms in cholangiocarcinoma. So these are all AMMF funded, or AMMF has funded at least part of these studies. And we were the first lab to generate an inducible transgenic mouse of cholangiocarcinoma. We've collaborated with Chiara on some of her work in Glasgow. And then we've got our own papers looking at things like Notch. We've had a long-term fascination with Wnt, and also how the immune microenvironment is established in cholangiocarcinoma, particularly as you transition from a pre-neoplastic setting into a neoplasm and a cancer. And I focused on Edinburgh because that's where I am, but the reality is we are not very far from Glasgow, so it's about 45 minutes away. And we collaborate very closely with Chiara, Tom Bird, Jen Morton, and Jeff Evans on cholangiocarcinoma. And earlier this year, Cancer Research UK awarded multiple centre kind of status to varying places, so Cambridge, London, Manchester, etc. And we got a Scottish one. So, and this is a formal agreement between Edinburgh University and Glasgow University that we will collaborate on cancer research, and we will work together as a kind of single institution known as the CRUK Scottish Centre. And this comes with about 12.5 million quid of funding for various kind of projects and infrastructure. What I wanted to point out, though, is that amongst all of this, we're the only one who has a defined liver profile and liver program. So we are being really proactive in trying to move liver and hepatobiliary into the research setting, and hopefully this collaboration between the two centres will accelerate what we are able to do. And so what are the aims for the hepatobiliary theme? You'll have noted from this slide that there's a variety of kind of themes within this. So colorectal cancer and liver are the largest two. But within this liver cancer or hepatobiliary cancer theme that is run by Tom Bird, myself, and Chiara Bracconi, we have three major aims. We want to find ways to identify cancers earlier across the spectrum of liver cancers. So this could be circulating biomarkers, it could be better imaging, it could be more refined diagnosis at the point of biopsy. We have a second aim where we want to develop our understanding of cancer genetics by modeling these. So Chiara does some beautiful work with these human organoids. We've very much focused on mouse models, and maybe, kind of we'll move into the fish because they're very genetically tractable. And our final thing is actually, and I'll talk in a moment about some of this. We are very good at identifying candidate mutational signatures in cholangiocarcinoma, particularly in intrahepatic. But that's probably because actually it's quite easy to sequence DNA. And it's relatively cheap. The issue we have, of course, is that for many patients there aren't actionable mutations. So how do we develop treatment profiles beyond genetics? And is that modulating the immune microenvironment? Is it modulating the stroma? Maybe it's modulating the epigenome, so the kind of modifications that cancers make to their own genetic material. And what we hope over the next 5 years is that we'll have better and earlier diagnosis of patients with hepatobiliary cancers, that we'll have more treatment options for those patients where we know the mutation, but also for those patients where we don't. And also from a fundamental level, understand how cancers form in the liver, particularly in patients who are high risk. So those patients like PSC patients who potentially will develop tumor at a much higher frequency than the general population. So those are our aims. 5 years' time, hopefully I'll be able to come back and tell you that we've met them all and would have made some headway. But that's where we're kind of going in Edinburgh. And I will just go straight on to the next talk. Sorry about my back-to-back.
Thank you, Luke. Just take us straight on to, I think, using patient-led genetics to define new therapeutic targets in intrahepatic cholangio. Thanks.
Thank you. So, still here. I've actually changed the title because actually of some of the work from Liverpool, I frantically changed my slides yesterday because I think we've got something quite interesting that we could collaborate on. So I thought I'd talk more broadly about defining new therapeutic targets in intrahepatic cholangiocarcinoma. And when we kind of started doing this, probably about kind of 6 years ago, we kind of were in a field that where genetic profiling was fairly immature. There were lots of papers with relatively small numbers of patients. It was quite variable, so you had lots of hilar cancer combined with lots of, well, not that much hilar cancer, a bit of hilar cancer combined with lots of intrahepatic. It was fluke-associated or it was spontaneous. All of these things were treated really as a kind of a single agent. And that caused a lot of problems because you lose resolution when you combine all those kind of variables. And it's very hard then to identify actionable mutations within that kind of patient group. Though that said, some did come up. So things like P53, which we've heard about yesterday and today, the IDHs, BAP, PBRM1, KRAS was very common. But still, even though we found this kind of consensus group amongst the patients, they really only occur in kind of a quarter to maximum a half of patients. And the drugs that are coming through, so particularly now for FGF fusions, but also for IDH mutations, are good, but they really are only useful in a tiny number of patients. And they've been driven very much by these mutations being present in other cancers. So I think it's kind of worthwhile to understand the genetics a bit more, but also try and find approaches that perhaps don't rely on these very, very rare mutations. All of that said, we did start with genetics. So what we struggled to do previously, just because of the way that the studies were structured, was identify recurrent mutations. So those things that perhaps occur in most people with cholangiocarcinoma. And you can do this in large data sets. If you have breast cancer where there are sequencing studies of several thousand patients, you can say, actually, this mutation occurs at this frequency within a population. But when you've only got small numbers of patients in these studies, it's very difficult to say this is recurrent or this should be actionable. And so we decided to take an approach where we combined a number of studies to give us just under 300 samples. And instead of using frequency as a measure of tumor initiating potential of these mutations, we used a pipeline called Intergen. And Intergen is a bit more kind of, I suppose, clever than frequency alone. So what Intergen allows you to do is define whether mutations in a particular gene occur at a higher rate, for example, than across the genome generally. It asks whether those mutations are at an active site, so are they likely to be pathogenic? And using those metrics, or are they in a regulatory domain as well? That's another kind of important thing. And using those metrics, you essentially get a score for your mutation. And it will predict whether your mutation is likely pathogenic or oncogenic. And we were able to go through all kind of 300 or so patients, and we identified 96 candidate mutations with high confidence that we thought were kind of potentially tumor drivers. And of course, we found the really well-described ones, so things like the IDHs, RAS, P53, but we also found these suites of mutations in other genes that are involved in are already included in COSMIC, so this is a database of pathogenic mutations across cancers, but also found lots of novel driver genes, and actually ones that occur with reasonable kind of frequency. So this dot here in our novel group is FA2, and it occurs almost as frequently as IDH, but we'd kind of never picked it out before just based on frequency alone. But when we look at that data more closely, we find that for about half of patients, just over half, there are 2 to 8 mutations. So most patients have kind of 2 or 3. But actually, there's also a very large group of patients for whom we couldn't identify an actionable mutation, or indeed couldn't identify a pathogenic mutation. So this isn't even about action. This is about there are no mutations in 18% of these patients. At least that's what our data says. But in about a quarter, you only have 1. So we are trying now to figure out ways of defining what a cholangiocarcinoma needs in order to grow beyond these kind of mutational profiles. And so by understanding these pro-growth signals, we hope that we can limit the growth of tumors rather than kind of having to stratify patients based on genotype. And so to do that, we've generated a number of mouse models and tools, and in the spirit of collaboration, we are happy to provide any tissue or kind of resources that we've got if people want to access our mouse models, because it's taken a lot of time. And we made this essentially screening platform where we use genome editing in order to introduce all 96 of our patient mutations into the mouse liver. And what we were able to do was express a mutant form of KRAS, so the most frequent gain-of-function mutation in cholangiocarcinoma, with this library of mutations, and generate tumors. And when you do that, you can identify, you can ignore the blue bit here, but you can identify which mutations are able to interact with mutant RAS to drive tumor formation. So these ones are actually always associated with RAS, be it KRAS or NRAS, and down here are genes that are mutated in this screen just when you have KRAS. And this allows you to do lots of things. So it allows you to understand the biology of RAS. It allows you to understand the interactions, the functional interactions between different mutations. So in this case, RAS and P53, which obviously came up in the Hyler sequencing. But also you can identify potentially new mutations. So things like mutations in neurofibromatosis 2, which when you include this with RAS, so now all of these mice had a mutant RAS, and they either had loss of P53, loss of NF2, or loss of both. You can see that, unsurprisingly perhaps, loss of P53 and loss of RAS, an activation of RAS gives you a cancer, but NF2 is sufficient to also drive tumor formation. NF2 loss is sufficient to drive tumor formation. And then when you combine these things together, you massively accelerate the rate at which cancer forms within the mouse. So these mice have to be euthanized within 14 days because the tumors grow so quickly. And we were able to show that loss of NF2 along with P53 severely increases the number of tumors that form. There are lots of, I could give an entire talk about NF2, but I'm not going to because it's quite tedious. And so what we can use this model for, though, because it's very aggressive, is to understand the fundamental mechanisms of tumor growth by screening, essentially, these animals and these tumors for the signals that allow cancers to grow. And that's exactly what we did. So we were able to show that in this very aggressive model, with a very, very sarcomatide and invasive phenotype, that that PI3 kinase and Wnt are activated. So this is GSK3 beta, which is a part of the Wnt pathway. This is beta-catenin, and we can see that in our NF2 mutants, beta-catenin has less phosphorylation, or the ratio is reduced. So this shows that the pathway is activated. And you can see here, again, dephosphorylated beta-catenin, so active Wnt signaling. And you can use these mice and these tumors to understand things like what's happening to dysregulate this pathway. So we found that there was a large number of cadherins that are downregulated, but also negative regulators of the Wnt signaling pathway, such as NKD2 and Frizzle-related protein, and there's an upregulation of Wnt9b. So by understanding those aggressive tumor types, you can kind of start to pick out recurrent signals that are required to drive tumor formation. And what I should point out is that we were quite surprised early on with sequencing that things that are gain of function or activating in other cancers don't seem to be there in cholangio. So if you have a colorectal cancer, you are likely to have a loss of function mutation in APC, and that will activate Wnt signaling. If you have hepatocellular carcinoma, there's a group of patients who will have an activating mutation in beta-catenin, but that's also absent in cholangiocarcinoma. So there is only one mutation associated with Wnt signaling, and that's RNF43, which turns over the frizzled receptor. And that, again, is mutated in quite small numbers of patients. But what we think happens is that regardless of what mutation you have, you activate these pathways. And this is important because it means that you no longer rely on the genotype of your cancer to kind of prioritize your patient for treatment, potentially. You could give any patient with essentially activated Wnt and PI3 kinase, a Wnt or PI3 kinase inhibitor, and hopefully that would have an impact on their tumor. And that's exactly what we did, not in patients, but in mice. And we had this model where our aggressive cancer driven by NF2 loss allows stabilization of beta-catenin and activation of PI3 kinase, and therefore activation of AKT, suppression of apoptosis and growth. And this is a mouse model where we delete P10 and P53 specifically in the bile duct. So they develop tumors over about 8 weeks, and we can label those genetically with a jellyfish protein called tomato. And when you stain for tomato, you can see all this cancer in the liver. It's here in brown. When you treat these animals for about 3 or 4 weeks with a combination of PI3 kinase inhibitor pictillisib and a Wnt inhibitor LGK974, you massively reduce the amount of tumor that forms. And actually, we did another study where we can show that if you treat as single agents, you don't get as much effect as when you combine the two together. So what does this mean for Wnt inhibition as a therapy? Well, there are trials starting, and Juan is part of it, kind of looking at using Wnt inhibitors to treat patients with cholangiocarcinoma, and I think this is very exciting. At the moment, the inclusion criteria necessitates actual actionable mutations in things like RNF43 or an ospondin fusion. But actually, we think that these tumors develop very early on a reliance on Wnt signaling and potentially PI3 kinase signaling too, that work together to form this kind of permissive state for a tumor, for a cholangiocarcinoma to form. So actually, targeting those pathways together might drastically reduce tumor growth, particularly in patients where actionable mutations can't be defined. So where are we going next? We've worked on Wnt for a long time, and other pathways are available. So what came out of both our combined patient data and also our functional CRISPR gene editing screen was that mutations in Notch actually occur with reasonable frequency in these intrahepatic cancers. So this is a mutation in Notch2. And Notch is a membrane-tethered receptor which sits in the plasma membrane of a cell, so in this case, the cancer cell in blue, and it meets its neighbor, its cell next door, which provides a ligand known as Jagged. And when that happens, you get sequential cleavage of the receptor, which then goes into the nucleus and becomes a transcriptional activator. And you drive a number of genes, so things like proliferation genes, stem cell genes, and things like that. So we wonder actually whether, we have a project now looking at whether Notch drives intrahepatic cholangiocarcinoma, and it was very interesting to see Notch mutations come up in the extrahepatic group. And so what we've been doing is trying to use our mouse models again to define the Notch landscape in intrahepatic cholangiocarcinoma. And this is what it looks like when you use these transgenics. So the brown cells here are cells that have lost the two tumor suppressors that are our favorites, kind of P53 and P10. And then we give an inflammatory insult called thiacetamide here in TAA, and over time, we start to see the formation of these very, very tiny tumors that then over 8 weeks kind of grow into reasonably large cancer. And when you isolate these cells, because like I told you, they have this fluorescent jellyfish protein called tomato, you can pull them out because they're fluorescent and a mouse doesn't normally fluoresce. And you can do RNA sequencing on these, and what we found was that Notch2 is very highly upregulated at the transcriptional level, as is Notch1, and Jagged1 and Delta-like1, another ligand for the Notch receptor, are both upregulated too. And when we take that tissue again and do histology, we can stain for the protein form of Notch2 and in cyan here, and you can see that Notch2 is expressed very, very selectively on the tumor epithelium. But what we wanted to know was whether Notch signaling itself can accelerate this phenotype. And is it a driver of cholangiocarcinoma? So this is why I like mouse modeling, right? So mouse genetics is really cool. You can do crazy stuff. So we can delete these tumor suppressors, as I've already told you, but we can also express this Notch intracellular domain. So this is that part of the receptor that kind of goes into the nucleus and binds the DNA after the receptor cleavage. And when we do that, and we express it in a cell that has lost P53 and P10, and then we give our thiacetamide, we find that tumors form. They form very fast, they're bigger, and there are more of them. So we think that in this kind of inflammatory system, the activation of the Notch pathway is additive to P10 and P53 loss and can drive acceleration of tumors. But is it sufficient? And that's a real big question, right? So it's not that often that a patient will have tumor suppressor loss plus an inflammatory insult plus Notch activation, blah, blah, blah. You know, we're adding things intentionally to try and drive these cancers forwards. But there are gain-of-function mutations in Notch2 in these patients, in Notch1 in the Hyler patients. And what we wanted to know was, without inflammation, is Notch enough to drive tumor initiation and growth when you lose P10 and P53? So here we use the same animal line, so we delete P10 and P53 in the duct and label them with tomato. And we use our tissue slicer and then clarify our tissues to get nice 3D images. So it's a brilliant machine. What you can see after about kind of 8 weeks or so, in a mouse that just has P10 and P53 deletion, so tumor suppressor loss, you don't have any tumors form. So these cells are very happy just to sit there and do nothing. You know, the loss of two tumor suppressors does not in and of itself drive tumor formation. But when you combine this transgenic where you put Notch intracellular domain in, within 8 weeks, you start to form these tumors. And very interestingly, they are a collection of very small kind of small duct cancer type things. But recurrently, we see these very large kind of IPNBs or bilins, these lesions in the large ducts, and they invade into the lumen of the large duct and occlude it. We're not entirely sure what this is. We've only had this transgenic line going for a few months, so we're still trying to figure all this out. But the reason I put these slides in is I think it's very interesting that maybe what you're seeing in your exome sequence or your genome sequencing is, you know, similar to what we're seeing here. You know, maybe we are in large ducts. Kind of there's a sensitization to Notch activation. I don't know, but it'll be very cool to try and figure it out. So we now have two kind of goals, which hopefully we'll be able to kind of update you with next year. So what we want to know is how Notch activation is promoting cholangiocarcinoma formation. So what are the mechanisms? What is that intracellular domain binding to? And you know, how is that achieving tumor initiation and growth? And can we specifically target notch receptors for therapeutic benefit? So this has been fraught in the field with problems. Notches required for things like your intestinal stem cells, your hair follicle stem cells, but there are some really, really good high-affinity therapeutic antibodies that have been produced over the last few years, particularly by Christian Siebel's lab, and those are now in clinical trial, particularly an anti-Notch3 antibody, but there are Notch1 and Notch2 antibodies. So I think this is what we're going to try next. So can we inhibit this pathway to limit the progression of disease, or at least the initiation of disease? And so with that, I think you've all had enough of me. I'd like to thank Scott, who did a lot of the early work on this model, and Annabelle Martinez-Lyons, who took on the Notch work. The Wnt work was very much done by Molly Wilson, who I have savagely deleted from my list because she's now finished her PhD. But she was AMMF funded, and we were very grateful, of course, to the continued support of the AMMF, because without them, we really would have never started lots of this work. So with that, I'll say thank you.